Good morning. So the way World Cup this FIFA World Cup is going, right? Pretty interesting. See, that's I was thinking. Uh, the way Morocco played, right? The way Morocco played and they beat that Portugal because Ronaldo was so favorite, right? So he's gone. Over there, the Croatia beat Brazil, right? That was before that. So these big guns they are out. And even in that match of that France versus England, you must have seen that, right? There was a golden chance for England for that penalty, and he hit it over the net, so he's gone. And Argentina, well, again, that was against that Netherlands, Germany, right? So even that was a good match. So now it is between Argentina and Croatia and France and Morocco, right? Pretty interesting. I don't know how many of you really follow it, but all these matches, there was so much to learn from these matches. Seriously, very interesting. By the way, I got few emails and Argentina, yeah, Argentina, even France, right? I'll bet even on France. They are the champion, right? Last time's World Cup champion, they are the France. Because that was a very nice match between France and England. So let's see. But seriously, now I won't comment. So someone asked me about who is your favorite. I said Brazil, right? They were out. On this day, someone asked that out of Morocco and Portugal, I said Portugal. So now they are out. So now I'm not going to speak anything about any team. Okay. See, I got few emails after that Saturday's session. Yeah, Rahul, exactly. And so if this thing happens तो तो obviously people will come to कि आप बोल दो कि ये कौन सी टीम जीतेगी and then they are out. Okay, so one email was, yeah, Ishan Kishan's 200 runs. Chris Gale's record was broken by 12 balls, right? That was amazing. Unfortunately, I couldn't see the match, but I saw the highlights. The real match must be similar to like highlights only, right? 10 sixes and so many fours. I like that statement when he said that say in 35 overs I was at 200, right? I could have made 300, right? That's the confidence. That's the confidence. And one of my friend forwarded me one very good article and छोटा पैकेट बड़ा धमाका, yeah. That's right. That's right. CR7 out for half time. Very true. Very true. And see, at times it happens like there is so much of overconfidence in some teams, right? But when you see the way the opposite team, say same thing is in Morocco because Morocco became the first African team to enter into semifinals, right? Because up till this point, all the teams, those who were from Africa, they were knocked out in quarterfinals. First time it has reached to semifinals, and look at their performance, right? Dedicated. So yeah, I'll agree to that, Deepak. Portugal was hyped too much because of Cristiano Ronaldo, right? But even same thing happened even in this case also. Na Brazil, the way Croatia played, that was outstanding, especially that penalty shootout. So what I was talking about was that email, one email. And when this article was sent, so I thought that out of that entire article I should really say one point to you, and that is, see, when we talk about self-respect, why this is so important, because this is the article from JRD Ratan Tata, right? And Mr. Ratan Tata There were several points, but one of the point is that no one gives a damn importance to your self-respect. No one cares, right? It is like you have to prove yourself. Yeah, this is so true. You have to prove yourself. And this is in reply to that particular person. That is when he said that एक बार डॉक्टर बन गए फिर तो मजे ही मजे हैं. No, not at all. Not at all. Don't be under that impression. Say in medical science, you are be ready to be a student for whole life. For whole life. Though human body is not going to change, but there will be so many new things coming constantly. So be, and as such I'm telling, whether you are a doctor, whether you are in medical science, or you are in any of the field, if you remain as a student for your entire life, you'll progress a lot. Seriously, you'll progress a lot. Yesterday, say it was Sunday, so what I did is I'm into RC cars also, remote controlled operated cars. Right. Very soon I'll be posting a video. I got the Traxxas, right? It is called as the Traxxas Jankis. And it's a car, very effective, very powerful. It goes almost at the speed of say 70 kilometers per hour, right? So I'll take a video of it and I'll post it. You'll love it. After quite a number of days, because all these things they use those lipo batteries, lithium polymer batteries, and you need to charge them very properly, which is called as a lithium polymer balance charger. Right? So yesterday I spent some time for doing all these things, right? And say now that car is ready. So maybe tomorrow or day after tomorrow I'll take it in out in the field. I'll take a video and I'll post it. Right? You'll really like it. So coming back to this, what I was telling was that the real life is very different. Seriously, real life is very different because in real life there are no vacations, and the most important thing is that it is only in our school there is consolation prize, right? There is a consolation prize. In real life there is no consolation prize. It is either you are a winner, you are successful, or nothing. No one knows you. And the best part is, as this topic, there was like so many emails they were about it. Okay, few seniors or few other students or colleagues they say kare itna mat pad. No, I would say never mock to those students who are studying properly, right? Those who are really studying properly, because who knows, today in pe has rahe hain, tomorrow we might have to work under them, right? So they are the real powers, right? So you be that power, right? So let's start with our today's topic. Tomorrow is the first semi-final, right? Tuesday. Tuesday. Today is Monday. So let's see how things go. So today's topic is cell membrane. Now, as such, we have talked about the cell membrane, right? So things which we have already discussed will go a bit fast, but I would like to add some extra things because this would be the foundation for our entire week. Because this week the plan is to go for all out for receptors, right? In detail about the receptors. And in this case, in receptors, I would be going bit overboard, right? Something which is needed in first MBBS will be going even beyond that. Seriously, will be going beyond that. Because at any point you have to learn, right? And you are smart enough to understand everything. And wherever there will be some new concepts, yes, we'll discuss it, right? But I want that you should be master of all type of receptors, because the moment this will happen, it will reflect in your not only physiology, right? You will be very comfortable in your pharmacology. You'll be very comfortable in the sign symptoms of medicine. This will improve your diagnosis, right? And all on top of all of them is you will be very happy, because when it happens, like you can visualize कि, अच्छा, this is what is happening in the body, right? One medicine, right? And this is what really happens when we give second medicine, how smartly it is eliminating certain areas and only pinpoint it is acting, right? So those concepts, this this is what I wish to go for this entire week, right? And yes, sodium potassium pumps will come, right? Again, we'll be watching that how they are practically used. Regarding the cell membrane part, we said that it is made up of bilayer phospholipid, right? Bilayer phospholipid. So those things will start going bit fast, and then we'll go for the specific things. So three things always remember, right? It is like proteins, lipids, and fat. Yes. Pharmacology will be in this week, means what I mean to say, pharmacology, pathology, even medicine, the diagnosis, physiology, everything would be integrated, right? Everything would be integrated. No, Jaydev, never do that. See, regarding that revision pattern of isotonic, hypotonic, and hypertonic, but then mistakes done 2-3 times, then confusion occur. No, there should not be any confusion, right? There should not be any confusion in those topics, because that is a very vital topic, very vital, very important. So if you understand the concept, you won't be making any mistake. Seriously, I'm telling you, you won't be making any mistake. If the mistakes are occurring, go back, right? And be very specific that where exactly you are making the mistake, right? Revise it, understand it, and then keep repeating. But yes, regarding that topic, regarding that fluid transport of fluid, that is very important. This topic would be very important. Then action potential, which will be understanding in the form of skeletal muscle, smooth muscle, and the cardiac muscle, very important. Because once you understand that, then the moment I say this particular drug will stop this action, immediately you will understand, fine, perfect, this is what will happen. I won't have to explain to you, in those things will appear in front of your eyes, right? So that's why this week is critical, is very vital. So yes, all those topics, please, any problem, do send a mail, right? Yesterday I have replied so many emails. Okay, so regarding this protein, very important. Almost 55% of the cell membrane is made up of protein, right? Lipids, they are about 40%. And that carbohydrate, I wrote fat, carbohydrate, it has to be carbs, right? Lipids and fat, they are same. Carbs, right? Carbs are about, say, 5%. ठीक? So that's the importance. Now, when we say that this is bilayer, right? So we drew it like this, right? Quickly, we used to draw it like this, right? This area is also called as polar, right? Polar means it is charged, right? It is charged. There is some charge, negative or positive, we'll talk about it, but it is charged, it is polar, right? And this is like external cellular fluid, ECF, and this would be inside the cell, ICF. This particular area, right, this is water soluble, correct? Hydro this water, and philic, this is water loving, right? It is this in between, this is what is hydrophobic, correct? Hydrophobic, right? And because this is fatty, right? So that is the fatty acid, and that's why this is the, also we can say, lipophilic, right? It loves lipids. So anything, if it is So anything, if it is lipid soluble, it would pass. It would pass through this membrane. This is our cell membrane. So we write one important concept, that is whenever we'll be learning the pharmacology, we'll be telling one thing, that what is the molecular weight of this particular substance, is this charged or neutral, and is this lipid soluble or not, and instantly we'll get the answer. So our concept, first concept which is coming up for today, concept, and you will remember this, right, that these things, they can cross the cell membrane. They can cross the cell membrane. Who can cross the cell membrane? One, whosoever is lipid soluble, right? If it is lipid soluble, yes, bindas, they can cross the cell membrane, right? Second, if it is got small molecular weight, right? Small molecular weight in traffic, right? Those who are on bike, right? They just pass through it, right? Big cars, they need proper clearance. Small molecular weight, or those who are neutral, right? Neutral means uncharged. Uncharged, because if they are uncharged, so then charged particles, they won't be affecting it, right? If positive they repel, positive negative they attract. So we'll be studying it in more detail, but these are the things, right? If they are neutral, they will pass through easily. So this is first concept. The second concept, the second one is, the second one is spatial arrangement is needed for, right? Spatial arrangement. And that's the arrangement which we'll see today. Spatial arrangement needed. And what's this spatial arrangement? Nothing special. It is like they are called as the channels. You require specific channels for whom, just the ulta of what we already talked about. One who is water soluble, right? They are not lipid soluble, so that's why we need a special path through which they will go, right? So those who are water soluble, lipid soluble, they can easily pass. Spatial arrangement needed to cross cell membrane, right? Just to complete it, to cross cell membrane. Okay, you know that, right? So water soluble, that was one. That previously it was the small molecular weight. Obviously these are the large molecular weight. Large molecular weight means what? Proteins, right? Large molecular weight. So big proteins, they'll be requiring some spatial arrangement, correct? And third, if they are charged particles, right? Charged particles, so they will also be requiring some spatial arrangement, and that is what is called as the channels. Do remember this, because this would be at many points, this would be the foundation for the pharmacology. Okay. Now let's say these channels, right? So these channels are like, say, like this. What these channels are made up of? They are made up of peptide chains, proteins, right? So these are like channels, and they are made of peptide proteins, right? Peptide. When we say peptide chains, that means proteins, correct? Peptide chains, that is protein. So their arrangement is like, say, like this, right? And then there is a formation of that entire chain. So things go here and it will come out, right? Say for example, for the water, right? It is what is called as the aqueous channel. Aqua is water, right? Aqueous channel. Now we go into slightly extra detail of this cell membrane, because now we'll be taking the clinical aspect of it, right? Clinical aspect of it. Sorry. Cell membrane, it has got what? Outer and inner, right? Because we said that there are two layers. So there is an outer layer and there is an inner layer, right? So that's the outer one and this one is the inner one, right? That's because there are two layers. Now in cell membrane, this upper half, that is the outer half, and then there is an inner half, right? So here we have got phosphatidyl choline, right? Now this is vital. Phosphatidyl choline, this is one, and sphingomyelin, that is what we see, right? And in the inner part, again it is phosphate, so phosphatidyl, but this time the name has changed, it is ethanolamine, right? Do you have to remember this? Yes, ethanolamine, because this is very specific, right? And very important, phosphatidyl serine. I have underlined it. It means where channels are present, there is absence of lipid. Lipid layer is there, but via that the channel has formed, right? So it is like just via that tunnel things go. So they don't have to worry about the lipid solubility. Right? Channels are embedded structures. Absolutely right. Channels are embedded structures. And yes, we'll be just within a minute, we are coming to that. But yes, you are right. Channels are passing through. So if this is like a cell membrane, right? So then channels, they are like this, right? This is the cell membrane. So all the solubility, etc., right? All the solubility, lipid solubility, charges, this, that, everything, it is affecting on this. But this channel, it is giving a pass through, right? But it is needed. Now this channel is very specific. If it is for water, it will allow only water. It won't allow anything else, right? It is not so कि चल मैं जा रहा हूँ तू भी आजा. No, it's not like that. Everyone has got a specific meaning. Only those channels which are for that specific purpose will be allowed, right? Not others. Acha. Now see, I underlined this phosphatidyl serine, right? PS. The importance of this is on the inner half, the inside. If it goes outside, so then there is chaos, right? Because then this cell will be marked as ke bas khatam. It is like a suicidal note going outside, and immediately it would be declared that this cell is not interested in living. It'll go for apoptosis. Apoptosis means kill, right? Eat me signal, it is called as eat me signal, and then all the factors of the annexin 5, etc., they'll start coming and they'll finish that cell. So this is what is called as the membrane asymmetry. Till this point we were under the impression that yes, upper layer, outer layer, and inner layer, right? We never discussed in that detail, right? We just said that it is the bilayer of phospholipids, but the thing is, the outer half is different, the inner half is different, and that is what is called as the membrane asymmetry, right? Membrane asymmetry. This membrane asymmetry tells that things which are should be kept inside, things which are for outside, they are to be outside. Another important factor is that when we'll be going into more detail, we'll find that those carbs, right, carbohydrates, they are present on the outer surface, right? We talked about it. Those carbohydrates, right, they are just hanging, but you won't find the carbohydrates on the inner side, right? They are present on the outer side. Right? This is one more difference. Regarding this phosphatidyl choline, sphingomyelin, phosphatidyl ethanolamine, and the phosphatidyl serine, all four are very, very important, right? Okay, we move to one another concept, right? Today there will be so many concepts. The thing what is called as the, if I say, right, and it would look a bit funny, but if I say that if this is one layer and this is the second layer, actually they move. Those layers, they really move one over other, right? That's true. They actually move. These phospholipids, right? These phospholipid layers, right? These molecules, they move laterally, right? They move laterally, they glide, they actually glide, right? And this is the property of fluidity, right? This is the property of fluidity, and that's why this is also called as the fluid mosaic model. Fluid mosaic model, right? This is for fluidity, and it is this fluidity which is vital for almost everything. If this cell membrane is, its fluidity is better, right? One simple example, it will improve your skin, right? And as the fluidity decreases, does movement affect channel also? Yeah, they affect, but channels will keep on working. These are molecules which are moving, right? So channels and everything will be there, right? But yes, they glide over each other, right? Let's see it in more detail and how exactly it really occurs. Who is responsible for this movement, right? Who is responsible for this movement? So let's say now I'm keeping some space between them, right? Because we would like to add something to it, and in between we add something. So friends, these are cholesterol. Cholesterol, right? It is this cholesterol. So these cholesterol, they are charged particles, right? And this entire model, the name, right? You should just know the name, right? This fluid mosaic model, right? That is by Singer and Nicholson, right? Singer and Nicholson. So just know the name. Now let's talk about, say, this fluidity. The factors which are affecting, right? Factors affecting fluidity. And within seconds, you will understand that why it happens. Like there are some changes we have learned in 11th standard. That's good. That's good. So we'll be adding something to that, right? So the temperature, right? Temperature is responsible. So as the temperature increases, this fluidity increases. Correct? Fluidity increases. What happens to the cholesterol, right? So what happens when the cholesterol is increased, right? When the cholesterol is increased. Now see, cholesterol was where? It is here, right? It is here. If the cholesterol is increased, right? If it is increased, what it will do? It will be tightly packed, right? It will be tightly packed. So when it is tightly packed, fluidity would decrease, right? Fluidity decreases. So when the fluidity decreases, this is the reason that why cholesterol, when it is so dangerous, right? In our food, in our food, right? When we say that fatty acids, correct? Fatty acids. Are all fatty acids bad? Well, not really. Not really. Fatty acids, they are divided into, say, two parts, correct? Saturated and unsaturated, right? Unsaturated. How decreases? Well, cholesterol, right? See, that's what I said. When the cholesterol is getting accumulated over here, right, it will be packed. This area would be packed. There would be very less space available for the movement. So relatively, once it is packed, right? Yeah, it will be in more pressure though. Yeah, that's right. There will be more pressure, so there will be, right? No, no, not it will be fast. In fact, when the things are packed, there would be very less movement, right? Things glide easily the things will be moving easily. There'll be more fluid if there is space given to that. If there is no space, if everything is packed, there would be minimal movement, right? So, because see, these are restricted areas, there are fixed areas. Now, in that fixed area, so the way we say that whenever there is monsoon season, right, those doors they just expand, right, and then there is so much difficulty in opening or closing of the doors, right. When there is no free space available, there won't be any gliding, and it that fluidity decreases. So that's what I said. It becomes tightly packed. When it is tightly packed, fluidity would decrease, right? Similarly, say these fatty acids. So those fatty acids, like say saturated, right? So saturated are like, so saturated are like what? They are like tightly packed, right? So saturated fatty acids, they decrease fluidity. This is a question which at times it is asked in many of the exams, right, competitive exams. And when it comes to unsaturated, so unsaturated, you draw it something like this, right? They are loose, right? These are loose, so they would increase fluidity. Regarding those palmitic and stearic acid, we are coming to that. But you got it? Saturated and unsaturated. So these unsaturated fatty acids, they are as such good, right? They are as such really, really, really good. Yeah, lack of double bond. Exactly, lack of double bond. Okay, now let's move on to these some special areas, right? Move on to some special areas. Amongst all different type of proteins, right? See, when we say, right, this is the cell membrane, this is the cell, right, membrane. Few of the three, 2, 3, 4, 5, 6, 7, right? That's our entire cell membrane. That's our entire cell membrane. Some proteins, right, which are passing through and through, these proteins, they are passing through and through 7 times, right? 1, 2, 3, 4, 5, 6, 7. Fatty acids, okay, we'll come to that, right? We'll come to that because it has to do with the transition temperature. So we'll come to that, right? Right now, I have just mentioned these things. We'll come to this why saturated acids and which saturated acids, right? We'll come to that. So first, I'm trying to give you the bird's overview that what exactly is there into our menu, right? And now we'll be going into the details. So it will come. So wait for some time. This is that protein which is passing through and through and through, right? It is passing through and through. So this is sort of 7 times it is passing. 7 times it is crossing the cell membrane. So that's why it is also called as the 7 pass receptor, right? It is also called as the 7 pass receptor. Right? Now, what is inside? Over here, there is a special protein, right? There is a special protein and it is called as the G protein. G protein. So this particular receptor, right, which has passed 7 times, it is basically married to this G protein, right, and forming a couple. So we would this so same protein, it can be called as G protein coupled receptor, right? We write receptors like this. So it is another name of it. So why to write this much long? Let's write it GPCR. GPCR. So when we say GPCR, that means there is a specific protein which is inside the cell, right? But outside, that cell membrane is getting crossed by those 7 pass, right? And because it looks like snake, so it is also called as the serpentine protein, right? All these serpentine protein or serpentine receptors, right? So all these things, they are one and the same thing, but they are telling us the characteristic. Now these are big molecules, right? They are big molecules. When we talk about receptors, right, the concept of receptor, as I was telling, that we'll be talking in detail about only the receptors, right, in this week. So what are these receptors? Simple. That's the cell membrane, right? Here is, right, here is something which is going through and through, right, which is going through and through. This particular protein, right now I'm just telling protein, right? This particular protein will be activated. It will say something inside only when a specific substance would come over here and talk, right? Say that in order to understand this, right? That's how I understood it. It happens, right, that in college, right, some specific person, right, we need not say declare it, but we for unknown reason we like that person very much, you know, right? So I always say that I'm explaining it from my own experience. So I understood it like that कि हाँ कि when जब वो हंस अगर जो उसने स्माइल दिया तो या कुछ होता है, राइट? अदरवाइज बाकी के कोई स्माइल दे तो कुछ नहीं होता. अच्छा, तो दिस मीन्स दैट it is exactly like this. This particular receptor, right, it is activated by a specific substance, right, and this is called as the ligand. Ligand means what? It is either a hormone, or it could be some chemical, or it could be some drug, right? But when that comes, yeah, something happens, right? बस तो these receptors are like that, right? लोग unnecessary फिर बदनाम करते हैं, but this is all natural, ठीक है ना? Say for example, and then the signal goes, right? It signal goes inside, and then there are कुछ कुछ होता है. हाँ, तो ठीक है ना. व्हाट्स इसमें हंसने वाली क्या बात है? क्या राहुल? See, you are the only person who is laughing. See, others are so serious. But no, I agree with you. So then say signals are sent. अरे, you too. Signals are sent inside, right? And then there are changes in the cell. ठीक है? And ठीक है, now don't think much. But this is what really happens. These are the receptors, right? Simple as that. And these, अरे हाँ हाँ ठीक है यार. Now focus. ठीक है. So signals are sent inside, and that leads to biological functions. ठीक है. This is the simplest of the explanation, right? That what exactly the receptor is. This is very, very vital. A specific ligand will only activate this entire process, not anyone, right? Say, for example, epinephrine, right? It can't enter, right? When we'll say, see the epinephrine, right? It can't enter the cell, but it can send the signals via receptors, right? So these are called as the receptors. Simple. What if substance can enter, right? What if substance can enter the cell? Because there would be few substances which can enter the cell. If they can enter the cell, then we will, because then they'll be generating their special effects inside the cell directly. So that's why we call them intracellular, right? Because they will be going inside receptors, right? intracellular receptors. Say for example thyroxine, right? It straight away enters into the cell, right? So we'll call them intracellular receptors because they can go inside. Done? So that's the concept which we understood. Now for these receptors, right? Now we'll write like this, receptors. When we say these pass-throughs, right? Pass-through means they are going through and through. So we can also call them integrated proteins, right? Integrated proteins. Integrated proteins means if we try to remove it, no, cell wall will be destroyed, membrane would be destroyed, but you cannot take it away, right? Not easy to remove. That's why they are also called as the transmembrane protein, right? They are also called as the transmembrane. All these words, when you'll be reading books, there'll be all these words because yes, emergency hormone. Transmembrane protein, sometimes in other book it would be written like integrated protein, right? They are all one and the same thing. So I'm trying to say make you comfortable with all those possible words, because when we'll be reading the books we'll find that there are different descriptions, right? So and this pass through protein, not easy to remove, right? We just can't remove it that easily. So not easy to remove, right? So this is one. Second, they are the attached protein, right? They are the attached proteins. So these attached proteins, they are attached either outside or inside, right? They are either attached outside or inside, and that's why it is leading to what is called as the membrane asymmetry, right? Membrane asymmetry, that it is not exactly what is outside is same as the inside. So these attached proteins, they are either outside or inside, correct? So what we'll call them? We'll call them peripheral protein, right? Peripheral protein. Now today, when we'll be talking about blood, we'll find that these peripheral proteins are so important because these are the proteins which are keeping our RBC like in its proper shape, right? If anything happens to these peripheral proteins, they lose this shape. When they lose this shape, they start breaking. When they start breaking, it leads to the entire sequence of, say, problems, right? So the thing is, these are the peripheral protein, and these proteins, they are loosely attached, right? They are loosely attached. It is not so that they are firm. Yes, they can be eliminated, they can be removed, right? So it means can be removed. Yes, they can be removed. Now how to remove it? Well, some proteins, they are extremely sensitive to pH value, right? So they can be removed. This will make that protein unstable, right? And some of the functions, or say with some ionic combinations. So this is where all those medicines will be coming into picture. So the idea is that integrated proteins, they cannot be stopped, right? They cannot be detached, but they can be stopped. Means simply, right? Instead of this, if this one is giving smile, right? No effect. Correct. Yes, it has to do with sickle cell anemia. Very right. So this was our self experience, right? बहुत कुछ होता है वाले, right? It has got no effect. So that is the specific ligand. And these are like attached proteins outside or inside, and they will make a big difference. Correct? Now, when we say integrated protein, right? Now, see, from cell membrane, now we are talking about what is attached to those cell membranes. So, one we already talked about, 7 pass receptors, right? 7 pass receptors. Sorry. There would be few in which some enzyme linked receptors, right? Which are not passing for the 7 times, but they are outside, they are inside. And then if specific enzyme comes and the effect starts, right? Someone will say कि चलो हाथ पकड़ के अंदर ले जाते हैं. So these are like carrier proteins. Right? These are like carrier proteins. There would be some other proteins whose function is structural stability, right? Just structural stability. So something which was like collagen and actin, right? They were there. So proteins for cytoskeleton, cytoskeleton, cytoskeletal stability. And the last one, there are few which will be just passing once. So in the way it was one pass protein, then there is like the 7 pass receptor, there would be some 1 pass receptors, right? So these are, but basically we just cannot remove any one of them. These are all very much integrated proteins. So when we were talking about cell membrane, right? When we were talking about the cell membrane, and in cell membrane, we said that about the fluid mosaic model. One very vital concept, right? And this is also like when I say that question asked in competitive exam, yes, actually asked, right? So I'll start putting the stars. So this star would indicate that it was asked somewhere in the exam. We talked about fluid mosaic model, right? Fluid mosaic model. And in this fluid mosaic model, we said that there were lipids, correct? And there were proteins. Proteins. Normally, right? Normally, we'll find that proteins are more and the lipids are less. So we'll say protein is more than lipids. And big difference, right? 55% and 40%. Right. Again, some spammer has come. रोज आ जाते हैं. Should take care of it. Correct. Huh. Right. So 55% and yes, 55% and 40% normally. But when it comes to, this is the exception, and they always ask you the exception, except no cell membrane, right? No cell membrane. Now in nerve cell membrane, you know there are two types, myelinated and non-myelinated, right? Myelinated and non-myelinated. So that when we really draw it like this, right? When we draw it like this, so it is this, right? Node of Ranvier, etc., right? So myelinated when it is present, non-myelinated when it is not there. So here we have to be very specific that nerve cell membrane which are myelinated, right? Which are myelinated, they have got the reverse proportion. In them, the lipids are much more as compared to proteins, right? There is here the 55-40 almost thoda same. This is 80% and this is 20%. So do remember this fact very crisply, right? Don't forget it. Definitely it was asked in exam, right? And worth asking a question. So when we say lipids, right, now we'll be getting that answer of all those, say, fatty acids. So lipids in cell membrane, right, in cell membrane. We divide it like this. The first one, phospholipids, right? Why it is so? I mean, because of this myelination is something which not only increases the speed, right, of nerve conduction, but it acts like a protection also. So this is phospholipids, right? These are like phospholipids. This is maximum, most abundant, maximum. In cell membrane, there are other type of lipids, and these are carbohydrate lipids, right? What we call as the glycolipids, right? So basically, they are carbolipids, and we can also call it like glycolipids. Glycolipids. And the third is our favorite, cholesterol. Cholesterol. And this cholesterol is basically precursor, right? It is precursor of vitamin D. Vitamin D is very much needed, and that's the reason that you should be getting sunshine. You should be getting the sunshine. You really check your vitamin D3, right? Vitamin D3. And you will be surprised that many times, even if we look so fit, but when we really check our vitamin D3, we find it painfully low. In the range of 10, 12, 15. Yes. And at times when you really see, they are like multi-millionaires, but their vitamin D3 levels, they are so low. Now there is no point in just eating calcium, because of the absence of vitamin D3, it won't be of any use, right? So that's the reason this is so vital, right? Sunshine is so much needed. Now, तो then is it so कि चलो दोपहर को 12 बजे जाके छत पे खड़े हो जाते हैं? No, it's of no use. Sunshine, right? It is specific, only specific hours, they are like that optimum range which are needed. Yeah, that's right. That's right. It leads to so many complications, right? Vitamin D3 and vitamin B12, these two, painfully low, right? Painfully low. So why morning exercise is needed? Because in the morning, when the sun rises, up to, actually it is said that up to actually say 9 o'clock, right? Up to 9 a.m. This is the sunshine which is the best, right? After that, so even pollution increases, and even this sunshine is not that effective, because then the intensity increases, right? It is like things are to be, it is what is called as the soothing sunshine, right? Not the scorchy heat, because scorchy heat would actually burn it. So this is why morning exercise is vital. It's so much needed, right? So let's name them. This is phospholipids, what we'll be discussing, right? So this is 1, this is 2, and this one is 3. So let's start with our first one. So just to orient yourself, right? We talked about cell membrane, and in cell membrane, right, we saw that there are two layers. And how can we check vitamin D3 normally in bone? Well, this is the lab test. It's a lab test. Just give your blood and they will give you the vitamin D3. Because if it is between 10 and 20, so it is like as if you are living in Somalia, right? Nutritional deficiency. It has to be more than 35, 40. But hardly you will get someone that good. And say there is no big rocket science. In case if vitamin D3 is less, right, all you need to do is, it is called as the cholecalciferol, right? Cholecalciferol. 60,000 international units, 60,000 IU. These are the capsules which are available, right? Take this depending upon your level, depending upon the level, per week, right? Every week start with 60,000 IU. Now this pack will come in the blister pack of say 4 tablets, right? There would be 4 capsules. So every month, this is to be completed in 1 month. And yeah, if it is dangerously low, so then we might have to increase the dose. Once the dose is properly maintained, once your level is maintained, so then even if you take about say 1000 IU per day, that is fine. That is fine. Huh. If your nutrition value and nutrition is good, but trust me, very rarely it happens like that our nutrition is so good that we maintain it properly. Right? Otherwise, so this D3 and B12 तो बर्बाद ही होते हैं. Just get it checked and you will come to know. It would be a huge surprise, right? When we feel career, I am so fit and why the level is so low. B12's biggest thing, if this level is low, you'll feel like by the end of the day, as if you are a knocked out boxer, right? Someone has beaten you so badly that you just can't move, right? बस सीधा बिस्तर में गिरे और मर गए, right? Literally that bad, right? Energy level drops to that painful level. शाम को किसी ने बोला कि ये कुछ काम है, नहीं नहीं कर सकते, नहीं पढ़ सकते, right? For lab tests, we need to take prescription. No need for prescription, right? All the labs, right? This is not US. In US, yes, you will need a prescription. In India, so things are very bindas, right? So you can just D3 and B12. And when you are going for B3 and D12, uh, what I'm telling, D3 and B12, right? So B12, D3, I would insist you should go for CBC also. CBC is complete blood count, in which we are very much interested in your hemoglobin and your WBC count. And do tell them that you want the ESR also, erythrocyte sedimentation rate, right? This thing will tell you is there any infection, and when the hemoglobin is there, why hemoglobin? In case of females, right? Suddenly, when the hemoglobin comes to about say 8 or 9, so then they are in panic. कि अरे इतना कम हो गया, है ना? However, lipid profile etc. So that is if it is needed. Otherwise, this much basic. Say your body is in good condition, right? It is in the best stage of life. So yes, all those reports if needed, lipid profile. So then you go for even the renal profile and all the details. But no, this much basic is needed. Why I'm telling for females hemoglobin, they should know their base level hemoglobin because their hemoglobin it keeps on shifting due to cycles, right? So this will give you a clear understanding that whether you should really go for say folic acid or not, you should go for iron supplement or not, because see iron supplement is needed, right? If the level starts dropping, हाँ, that if the hemoglobin is improper, it leads to many complications. But immediate complication is oxygen carrying capacity because it is the hemoglobin which is carrying the oxygen. If the hemoglobin is less, right, obviously the efficiency of the person will go down like anything. And the best part is that these tests they are not that expensive. 4-5 हजार रुपए के टेस्ट, no, they are pretty cheap, right? So phospholipids, first what we talked about, that outer one, right? This is phosphatidyl, right? Phosphatidyl choline, right? PC, right? Say you are medico, right? So phosphatidyl choline, PC is phosphatidyl choline, right? We are not from Bollywood. Okay? Can we predict women with the help of pulse oximeter? A rough idea? Well, you can, but see what happens. Pulse oximeter is oxygen carrying capacity, right? It is oxygen carrying capacity when you are at rest. So even if someone's hemoglobin is low, but when the person is at rest, so things are fine. Say in COVID, why it was said that someone would attach the pulse oximeter, are 97, 98, chalo, chalo, good. No, one of the test was, no, tell that person to do a brisk walk for 5 minutes and then check, and you'll be surprised when you check it, you'll find that this oxygen level is dropping like anything, right? Same thing is true over here. So when you go for a run and then you find that you are breathless, right? You are breathless, and then when you check your oxygen concentration, you'll be surprised that it is quite low. So when at rest, so yes, it will come normal, but less hemoglobin. So we can't say that pulse oximeter is giving us a very good prediction, right? Yes, it is telling that at that point of time, at that state, things are okay. Regarding this phosphatidylcholine, right, also called as the lecithin, right? Remember, and we are about to discuss one very nice concept of on this is the lecithin. What is lecithin or phosphatidylcholine? This is a superb thing which is acting as surfactant, right? Surfactant. What is this surfactant? See our alveoli, right? As things divide, right? Things divide. So that's our trachea, and then divides into right and left primary bronchus, and then they divide, divide. Finally, they will come to what is called as the alveoli, right? They are like alveoli. And these alveoli, their tendency is to collapse, right? They always try to collapse. In order to prevent this collapse, the inside there is release of one substance, and this substance acts like a stretch membrane, right? A stretch membrane, and this stretched membrane, and this substance is, friends, surfactant. Surfactant. So in case if anything wrong happens to surfactant, those alveoli would collapse, right? They will collapse. So this surfactant is lecithin. So in babies, if the lecithin is not proper, right? If the concentration of lecithin is less, nothing would happen because the lungs are actually collapsed. ठीक है ना? Lungs are actually collapsed because baby is not breathing, his lungs are not active. But when he'll take birth, at that point, if the lungs are immature, then rest of the life is painful. So that's why this lecithin is so important, because it is a major surfactant. It is a major surfactant. So we need to estimate for the lung maturity, we need to estimate whether this lecithin is okay or not. Right? So this is phosphatidylcholine or lecithin. That is the first one. The second one, what we said is sphingomyelin. Right? It is the sphingomyelin. This sphingomyelin, which we write as S, this is also a surfactant, but it is a minor surfactant. Right? This is not a major surfactant, it is a minor surfactant. It helps, but the major work is always done by lecithin or the phosphatidylcholine. Yes, that's right. It is the layer above alveoli, means inside the alveoli, right? Inside. This is the inside. Air goes here, right? This is the inside. Got it? It is inside. So that's why it keeps it stretched. So in obstetrics, right? In obstetrics, when the female is pregnant and you really want to check out whether the lungs of the fetus, they are properly matured, right? So this is the alveoli, right? And the oxygen goes inside, carbon dioxide, it comes out, right? Over here there would be like blood vessels, right? And they go for all these exchange. So yes, surfactant is here, right? That's the layer. So what I was talking about, that for fetal lung maturity, right? Now see, it's such a simple test but so vital. For fetal lung maturity, two proteins we talked about, lecithin, right? Lecithin and sphingomyelin, right? Sphingomyelin. So lecithin and sphingomyelin, this is, there is a ratio. This LS ratio should always be equal to or more than 2. So that means lecithin should be double than the sphingomyelin. Yes, they give steroid shots. That's right. So if the LS ratio is 2.0, that means lungs are matured, right? Lungs are matured. And yes, steroids they are given, corticoids they are given, so that it goes. Now this particular test, what it is actually, the amniotic fluid is extracted, right? So amniotic fluid, right, from the mother, it is syringed out, small quantity, it is put into test tube, and then it is shaken, right? It is shaken. And because both these proteins, they have got different molecular weight, so you will find that it, the bubbles will be formed, right? So there will be two layers. So when the bubbles they are formed, that means everything is okay, right? So this test is also called as the shake test. It is called as the shake test. It is also called as the bubble test, right? Bubble test. Or there is a one more scientific name, it is called as the Clement test, right? Clement. Do remember this, Clement test. So that is where we get the idea the whether it is proper or not. Now when we say sphingomyelin, we need to mention one important thing for sphingomyelin, right? Sphingomyelin is like minor surfactant, right? So in lungs, so it has got the role, but that role is not that crucial. It has got a major role in myelin, right? Myelin in nerve cells. And myelin is that which is acting like a coat, so it improves the speed, and at the same time it prevents the signal from dying early, right? So nerve cell membrane, so that is the sphingomyelin, very very important. We move on to our sorry. So we talked about two, right? We talked about one, and that is phosphatidylcholine. Second, we talked about sphingomyelin, and now we talk about the third one. Third one, which is into the inner layer, right? It is for the inner layer, and that is PS, or the phosphatidyl serine. This PS is strictly on the inner surface, strictly inner surface. That's true. That's true. Because at that point, lecithin is there, but because there is no air, right? So the moment air goes, and these surfactants they start releasing, and then they pop up all those alveoli. So alveoli pop up, right? They start opening it, and as the baby matures, right, they more and more alveoli they will be getting, say, functional. So this is inner surface, right? So when we have got this cell bilayer cell membrane, so we find that this PS is here. We don't find PS here, right? It is not there. This is extracellular fluid. This is intracellular fluid. So this is also called as the membrane asymmetry, right? Membrane asymmetry, different phospholipids on the outer layer and the inner layer. Now, if this goes out, that means if it appears over here, if this phosphatidyl serine appears here, so then straight away, this is the signal, right? It is the signal that, okay, eat me signal. It is called as the eat me. right? Eat me signal is nothing but it is called as the apoptosis, or the cell death, cell death. So it is like a red flag which has been raised by the cell that yes, eat me, finish me, right? This is also called, and the moment it happens, there is another cellular protein, right? Very important, right? This is another cellular protein which is already like a hunter, he is moving, right? It is annexin 5. So this annexin 5, the moment he watches this, immediately he comes and he gets attached to this PS, and that is what leads to formation of annexin 5 PS complex. Now why we are discussing this annexin 5 PS complex, right? Because when the phosphatidylserine is exposed on the outer side, then annexin would catch hold of it and would form this complex, and this complex can be seen. Yeah, it can be seen. If you really want to see, so then you require some special fluorescence coated substances, right? One of the very important, which is available in the form of a kit, right? It is called as the Alexa, right? Not that Alexa. Alexa Fluor, right? Watch for the spelling. This is for fluorescence. That's why Alexa Fluor 488 annexin 5 kit, right? This is a kit which is available, right? And this thing, with this, you can actually visualize that whether there is presence of annexin 5 PS complex. Importance, if this is positive, that means those cells, they are now going to die, right? They are going to die because it is an apoptosis indicator. It is called as apoptosis indicator. And when this is positive, this means this apoptosis has already reached to annexin 5 staging, right? Also called as the annexin 5 staging. Now all these stages, yes, we'll be discussing when we'll be talking about the apoptosis or the cellular death, right, in more detail. But this is just you should know. The next one, right, which is fourth one, which you must know, it is IP3. Right? This is phosphatidyl inositol. This phosphatidyl inositol, whenever it appears, remember this is for the hormones, right? It is pertaining to endocrinal system. So this is, you can say, it acts as a second messenger, right? Second messenger. Now what is second messenger? Second messenger means these are which are inside, right? These are the inside. So there would be, this is the cell membrane, and this IP3 is over here sitting silently till the message from the outer one comes, right? So there might be another receptor and signal is sent, and it affects, it tells IP3 that go and start performing some operations, right? Because it is affecting the endocrinal system. It is triggered by external factor, right? So that external factor would say to IP3, IP3 would go and will start doing some endocrinal system changes. So that's why this IP3 is also called as the second messenger, because this would be the first messenger. That ligand would be the first messenger where it will say, yes, let's start the process. No, this kit never helps to stop apoptosis. This kit helps to check whether the apoptosis has reached to this level or not. It just for the diagnosis, it is not at all for the treatment. Okay? It is not for the treatment. One more. This is another protein. It is called as the cardiolipin. Now, this is a very exclusive protein. We'll find this in mitochondrial membrane. Then what's its use? Its use is to diagnose, right? It is for the diagnosis. Kits are used for, say for example, use of X-ray. Use of X-ray, right? It is whether the fracture is there or not, correct? Because this is what is called as the diagnostic procedure, right? Many of the procedures, they are for only for diagnosis, they are not for treatment. Say for example, you do barium meal, barium swallow, you come up with that this person is having intestinal obstruction, or this person is having some, say, space occupying lesion. So it is for the diagnosis, right? You do ultrasound and you come up with that there is a mass. It is not going to give any treatment, but well, diagnosis is important because then and then you can proceed with the treatment, that what is to be done. So cardiolipin, right? Cardiolipin, which was extracted from, first it was extracted from human heart, right? But it is exclusive, right? But it is exclusive in mitochondrial membrane, right? You won't find it anywhere else, right? It is in mitochondrial, right? It is into mitochondrial membrane. Now we know that that must be the reason it has been extracted from the human heart, because human heart, those cardiac cells, they are having maximum number of mitochondria, right? Because mitochondria, they constantly keep on supplying. One person asked very interesting question, that from where mitochondria gets the power supply, right? The energy. For that we need to go into the details of the entire process, right? For mitochondria, initially it uses a ATP, right? It initially uses ATP and then it multiplies and gives it in burst, because initially it uses 2 ATP and then it gives 36 ATP. So yes, you need to invest something to get bigger returns. So initially it uses. So that's the reason that our body is so live, right? So what I was talking about, yeah, cardiolipin, and it is exclusive in mitochondrial membrane. Now, there are few disease. Now listen carefully. Yeah? There are few disease in which, yes, ATP is synthesis. That's right. So there are few disease in which those microorganisms, they form antibodies against cardiolipin, right? So these are antibodies against cardiolipin. And these antibodies you can detect, and that is used in, say, investigation of, say, some sexually transmitted diseases, right? It is called as the VDRL test, Venereal Disease Research Laboratory Test, VDRL test, right? Typically, you'll find that you must have heard of syphilis, right? Syphilis. So this is what is tested, because it creates that antibodies against cardiolipin, right? The whole name would be treponema. It is called as the treponema pallidum syphilis, and it is this which produces antibodies against cardiolipin, and that antibody, anti-cardiolipin antibodies, they are detected into this VDRL test, right? So it is like anti-cardiolipin antibodies, and they are tested in VDRL test. So this was our first group. Our second group, which was glycolipids. Now this topic is short. This glycolipid divided into two parts, right? One which is ganglioside. ganglioside, ganglions, right? And second, which is cerebroside. Both of them present in brain and in large quantity, abundant, so much, right? Large quantity. Regarding these ganglioside and cerebroside, more when we talk about, when we actually discuss the nervous system. Right? The third one, this was cholesterol, right? Now, this cholesterol, the third important type of lipid, and which we said that this is nothing but a precursor of vitamin D, precursor of vitamin D. You know? What it does, it maintains fluidity. Now, how exactly it really does that, maintains fluidity? There has to be some special characteristic. You see, there are basically two types, right? Two types. So one, those lipids which will increase membrane fluidity, and other one which would decrease membrane fluidity. ठीक है? Those who are increasing, they are good for health, and these are obviously bad for health, right? Simple. Those who are good for health, we said that they are unsaturated fatty acids. I remembered it like U means Uttam. Uttam means good, right? So they are good. And these are saturated. Saturated means sada hua, right? So saturated, so that we don't make mistake. So saturated fatty acids, right? So they are bad. Okay. So good. They are, there are very specific which are good, right? One is, and you'll see these things even on many of the labels or many of the ingredients of those food products, right? One which is called as the PUFA, right? This is polyunsaturated fatty acids, right? Simple. This is PUFA, very very vital. But then you'll find this word essential fatty acids, right? And this is question, essential fatty acids. So which are the essential fatty acids which are good for the health, right? And these are linolic and linolenic. Yeah, omega 3 is PUFA, right? This is linolic acid, right? This is one, and second is linolenic acid, right? These two are essential fatty acids, and one more, and this one is arachidonic acid. In many of the products you'll find presence of these fatty acids, right? Those which are decreasing the health or not good, these are two and must know, right? This is stearic acid, and second is palmitic acid. Stearic and palmitic, these two. Just check out on those wafers or all these packets that you will find that so many places stearic and palmitic acids, they are present. Seriously. Now, why they are? What's the difference? The idea is, the concept is transition temperature. transition temperature. In transition temperature, here is the solid and here is the liquid, right? There is a solid lipid and there is a liquid lipid. And as we said, that it depends upon this at what temperature things are getting converted from one state to another state, and that is called as the transition temperature. So this transition temperature, right, now the concept is coming up, that transition temperature, right, transition temperature is inversely proportion to fluidity. That means, think of, lower the temperature. If the temperature is low, right, lower the temperature, higher the fluidity, right? Lower the temperature, higher the fluidity. That's right. So, right, and higher the temperature, lower the fluidity, right? Simple as that. So we can draw it like, say, fluidity and the temperature. Okay? And the temperature. So higher the fluidity is at lower temperature. This is good, right? And that's what all those unsaturated fatty acids. Because at lower temperature they will change the state. But those saturated ones, right? Saturated ones, at higher temperature they will change its state. So they'll keep on affecting, right? And this will drop the fluidity. This is what is bad for us, right? So do remember this part, that how it really affects. Right? So finally, we come to our all-time favorite, cholesterol. Why this cholesterol, right? The cholesterol is that it is meant for, it maintains higher temperature is high fluidity. Where exactly? No, because when the temperature rises, right? When the temperature rises, let's check it out. Let's check. Let's go back. Because this is in reference to these fatty acids. So over here, saturated, yeah, that is because saturated is bad, right? So this is decreased fluidity. This is okay. Where? Acha. You are talking about this? Got it. Ha. So this is not in reference. See, this is in reference to what? This is in reference to the entire fluid mosaic model. Yeah, exactly. Got it. Now what you are asking. This is with reference to entire fluid mosaic model. One thing is fluid mosaic model, right? That is our normal skin temperature, right? Normal skin temperature. When the temperature rises, right, the fluidity increases, right? Fluidity increases as the temperature. This is about the temperature of the cell itself, right? This is when we are dealing with the cell membrane itself. So this is with relation to cell membrane. While what we were discussing was, that is when it comes to those saturated and unsaturated, that why they are good or bad, right? So that was in relation to the transition temperature, right? This is like temperature of our cell itself, right? Over here, this is about the transition temperature, right? Transition temperature is which is meant for those lipids, right? So that is when unsaturated fatty acids, why they are good, right? They are good because their transition temperature is lower, right? So they maintain the fluidity. Acha. Coming to this cholesterol, right? Cholesterol, when we say it maintains fluidity, right? It maintains fluidity, but till it is under control, right? When the cholesterol starts rising, when the cholesterol rises, the things become compact and the fluidity decreases. But the speciality of cholesterol is that it neither increases nor decreases with the temperature. The cholesterol himself, that's why it is also called as the fluidity buffer. Fluidity buffer. So it maintains that fluidity, but its proportion should not increase. Right? And what is the real use or the importance of this fluidity? The one classical thing about the lipid is, see, one, that what we said, that it keeps that skin or the tissues fluidity. There is membrane asymmetry, right? That's what we even saw, that what is inside and the outside, they both are different. And the third is, have you seen, at times we do biopsy, what is called as the FNAC, fine needle aspiration cytology, right? It is called as the fine needle aspiration. Aspiration means to take out, cytology, because we'll be sending this material for histopathological examination to check the presence of specific pathological cells. So a fine needle is put, right? And then it is like trocar, you just cut it and then you take it back. So the tissue comes out. But what about that when that needle was introduced, right, via the skin? So what about this puncture? It reseals, right? It reseals. Resealing, that is due to this lipid property, right? Where things, they reseal. So that's the interesting property of it. So I think we stop at this point, right? Tomorrow we'll be taking this thing to next level, right? Where we'll be discussing in more detail about the receptors. So what would happen tomorrow? We'll be going into the details of straight away receptors. And in that, we'll be talking about all various types. Supervision, we do that. We'll do it. So for this is for tomorrow, right? So we'll talk about that those receptors, what we talked about, that some are transmembrane, right? Just a brief transmembrane. So you now know transmembrane receptors very well. Then there would be few peripheral. Peripheral, now you know that peripherals, they are like either outside or inside, right? Very clear. And one very interesting type of protein, which is called as the lipid anchored, right? It is like lipid anchored protein. Lipid anchored protein means these are the proteins which are not directly connected, but they require something, right? Okay, this is a big topic. We'll talk about it. Just let me finish this part. If I want to show this thing graphically, right? These three types. So when I draw something like this, this is cell membrane, correct? This is cell membrane. So this is our first type of protein, the transmembrane. ठीक? Then this is like our second type of protein, which is peripheral protein. And say for the lipid anchored protein, we draw something like this. Like this. Say this is IP3, ionositol phosphate, right? IP3, ionositol. inositol. And then there is a protein, right? So this is the third type, that is lipid anchored. Tomorrow you have to remember just this much, right? So this one is integrated, this one is integrated, and this one is peripheral. This one is peripheral, right? Bus. So now the entire classification will go accordingly, and then we'll be talking about, say, our sodium potassium pump. Tomorrow we'll be discussing about even sodium potassium pump, right? Because say this integrated protein, we also call it GPCR, G protein coupled receptors, right? G protein coupled receptor, because at the end over here there is a G protein, correct? There is a G protein. Similarly, that sodium potassium pump, sodium potassium ATPase pump, so which will be pushing 3 sodiums out and taking 2 potassiums in. So that is also a channel, right, which opens and closes, but it keeps on working, ubiquitous, right, present everywhere, non-stop works. So this is like pump, right? So this is like pump. Pump means it is constantly working, throwing out, taking in, throwing out, taking in, right? Or there would be few channels. So those channels, they are present, but they will be active under certain conditions, right? And those conditions, it could be, say, voltage, it could be temperature, it could be some specific substance. So that is what ligand is there, right? Who is giving smile, right? That's so important, right? ऐसा तो ना है कोई भी smile दे तो. So that's what we'll be talking. And when we'll be talking about, say, peripheral proteins, we'll see that how these proteins which are on RBCs, how those proteins which are on skeletal muscle, right? Test we can take test on, say, Saturday. Let now that there would be sufficient material, right? So we'll take a test. Sure, we can take the test on Saturday, right? Declaring from today only, so that everything whatsoever was discussed in physiology, because this would be the physio test, right? Physiology test, right? From the day one till the Friday, whatever we discuss, we'll take that. Perfect, good one. So these peripheral proteins which will be responsible and helpful for this our RBCs, right? That's what we'll discuss. So that is for tomorrow, right? And yes, there would be few very interesting things about, say, how the glucose transporter acts, etc., right? So all those will be for tomorrow. Let's go for a high speed revision. Okay, and let me save this file. Done. So Argentina versus Croatia and France versus Morocco. Okay. So cell membrane, bilayer phospholipid proteins, lipids and carbs all are there. And in cell membranes there are, say, receptors, and knowing this will help us in physio, pharmac, medicine, diagnosis at so many levels. Important is that this outer one is hydrophilic, right? Both ends, they are hydrophilic, and in between area, that is the lipophilic, it loves lipids, right? Those are the fatty. Important one point is polar, right? Polar, or they are charged. So that's why any charged particle which will come over here, either it will stick or it will be repelled, but it won't be allowed to pass through. That's why uncharged particles, they will be going more easily. So this is the concept. Who can cross the cell membrane? Lipid soluble, small molecular weight, and neutral or uncharged. Because charged one, either they will stick or they will be repelled, right? But they will not be allowed to pass through. So this is what. So that's why special arrangement needed in the form of channels to cross cell membrane. Water soluble, right? Large molecular weight, and the charged particles, they won't be able to pass through. That's why special channels, special pumps, that is what is needed. And all these channels, pumps, they are made up of peptide chains, very specifically, right? Cell membrane, outer half and the inner half. In outer half, remember this carbohydrate, right? These are important names which we discuss. This is phosphatidylcholine, right? Sphingomyelin, phosphatidylcholine, also called as the lecithin, right? As we saw, phosphatidylethanolamine, phosphatidylserine, very vital, right? This is what is called as the membrane asymmetry. Asymmetry means outer and inner are different. Phosphatidyl and now the phospholipid molecule, they move laterally, they glide, and that is what is called as the fluid mosaic model. Cholesterol, they are in between, right? This model was given by Singer and Nicholson. Factors affecting fluidity. That is, as the temperature increases, temperature of the surface increases, fluidity increases. Right? It is because of the expansion. Expansion. Increased cholesterol, that becomes tightly packed, right? And that will lead to decreased fluidity. Decreased fluidity also means decreased flexibility, right? That's why cholesterol is dangerous, especially when it starts getting accumulated. That's why cholesterol level is to be kept under control, right? That's why exercise is so much needed. Fatty acids, saturated and unsaturated. Saturated, they are bad, they decrease fluidity. Unsaturated, they are good, they increase fluidity because of that double bond. Special areas, as we said, that this is the 7-pass receptor, G protein coupled receptor, or GPCR, or serpentine receptor, all same. Well, this one is, right, our ligand, right, could be hormone, chemical, drug, and because of this ligand there would be signals which are sent inside which will lead to the biological functions, right? And substance can enter into the cell. Well, in that case we call it intracellular receptors because they can go directly, and that is like, for example, thyroxine. Receptors, they are like pass through integrated proteins, transmembrane protein, and they are not easy to remove. Attached proteins, they are outside or inside, right? Peripheral proteins, loosely attached, they can be removed either with a change in the pH or by the ionic combinations. Integrated proteins, they are 7-pass receptors, then enzyme linked receptors or the carrier proteins. Tomorrow we'll be going into the details of these proteins, right? The complete classification. For cytoskeletal stability or the 1-pass receptor. Cell membrane, right? That is fluid mosaic model, lipid and protein. And in this, normally proteins are more, right? Proteins are more as compared to lipid, except the nerve cells, right? And that also in nerve cells, we talk about the myelinated one, in which lipid is extremely high, almost 80%, right? As compared to the protein. In the cell membrane, we have got the phospholipids, we have got the carbolipids, and we have got the cholesterol, right? These are the three groups. Phospholipids, over here we talked about vitamin D3 and how the cholecalciferol or vitamin D3 60 kIU international units per week, that is what is to be taken. 1000 IU, 1000 to 2000 IU is like a maintenance dose, right? Phospholipids, phosphatidylcholine, PC or the lecithin, and it acts as a surfactant. It is a major surfactant. Surfactant is something which is preventing collapse, right? It prevents alveoli collapse. Alveoli collapse. Sphingomyelin, S, this is a minor surfactant, and surfactant is something which is acting like a stretch membrane. And in obstetrics, for the fetal lung maturity, you need to go for LS ratio, right? It is called as the LS ratio. This LS ratio should be more than or equal to 2. So that is the indication that lungs are matured, right? The amniotic fluid is taken, shaken, and then you see the level. This is also called as the shake test or the bubble test, or the name to be remembered is called as the Clement test, right? Sphingomyelin, minor surfactant, but another thing that it is present into the myelin sheath in nerve fibers. Phosphatidylserine. Phosphatidylserine is strictly for the inner one, strictly, right? And as it goes outside, right, as it goes outside, it is like eat me, right? It is like eat me signal. it may signal, and that is apoptosis or the cell death, right? And that is where this annexin 5 cellular protein annexin 5 comes, and it forms this complex, and it can be seen by the kit Alexaflor 488 annexin 5. This is nothing but fluorescent material, right? Fluorescent material. So it is also called as the apoptosis indicator, that is the annexin 5 staging. IP3 phosphatidylinositol, this is acting as a second messenger because it is inside, right? It is inside. So when the message comes from the top, this IP3 is now into action, and it starts affecting the endocrinal system in the form of hormonal release, right? Hormonal release. So these are for the diagnosis. Cardiolipin, which is exclusive into mitochondrial membrane. First, it was extracted from the human heart. So that's why cardiolipin, right? Microorganisms, antibodies against cardiolipin. Say, for example, that microorganism, Treponema pallidum syphilis, when it affects that, it produces antibodies. These antibodies, they are detected by this VDRL test, right? Venereal Disease Research Laboratory test. Glycoprotein, ganglioside and cerebrospide, classically in brain, huge quantity, right? Cholesterol, finally, precursor of vitamin D3, maintains fluidity. Two types, right? One which improves the membrane fluidity, second which decreases. Improves, good one, it is this PUFA, right? Polyunsaturated fatty acid, and the essential fatty acid in the form of linoleic acid and the linolenic acid, and finally the arachidonic acid. On the other side, bad are stearic and the palmitic. The reason behind it is the transition temperature. Temperature it changes one state to another state. So this transition temperature, lower, it is inversely proportional to the fluidity. So lower the temperature, better the fluidity, right? And that's the reason it goes like this. That is, in unsaturated fatty acids, it is lower temperature, and in saturated, they have got the higher transition temperature. Cholesterol neither increases nor decreases due to temperature, right? That's why it is also called as the fluidity buffer. So fluidity, membrane asymmetry, and the FNAC, fine needle aspiration cytology, where it takes care of the resealing, right? So that's our, say, this is about our receptors. The first type of receptor, that is the integrated one. The second type of receptor, which is a peripheral one, outside or inside, right? And the third type, which is not directly attached to the cell membrane, it is connected via IP3, right? That is inositol. In our integrated one, this is a GPCR, that is G protein coupled receptors. There would be the pumps, right? All those things which we'll be talking about, yes, tomorrow. And yes, on Saturday there would be physio test. Yes, you got a question? Yes, go ahead. I saved the file. I'll put it. Send a mail. Send a mail. Right? You can send a mail to me. So then conveniently I'll answer that about this PG, etc. Okay, so thank you so much, and we meet tomorrow, right? Same time. And tomorrow we'll be talking about the game of receptors. Thank you and bye bye. And I'm putting this file into our folder, and then I'll send you the link. Thank you. See you tomorrow.